He has, as you know by his resume, he has experience in productivity, molecular biology, entomology, environmental effects on plant growth, and sustainable horticulture. And today he's going to talk to us about the adaptation to acclimation using papaya's unique biology to improve productivity. Dr. Vincent?
Thanks everybody for coming. Can you hear me okay?
Yep.
All right, awesome. So so my original background was in journalism, and I just realized today I broke a rule of print journalism, but I won't tell you what it is. It's on this slide. Anyway, so just a quick overview of my talk. Is this okay with the microphone with me moving?
Yep.
Okay, great. All right, I'll just give very briefly what my background was before coming to work on my PhD, and then I will basically dive into what my dissertation projects were and talk a little bit about their implications for papaya production, but then also for horticulture more broadly, and then conclude with basically what I would hope my approach to be in this position where I selected. So just very, very briefly, basically when I started in horticulture, I started working in blackberry, did a number of different things. We ended up working on pest management and flowering phenology. Went on from there to work very closely with the growers in the state who were facing some planting decline problems like what you see right there, and trying to come up with some diagnosis for what was causing those problems, as well as some alternatives for management so that they could basically restore their growths or their plantings to production. And eventually that actually led more recently to some work with Driscoll's, trying to basically condense the existing academic literature into information that was usable for their producers and nurserymen. So now I'm going to switch gears. We'll go into papaya. Normally I talk a little bit more about what papaya is, what it looks like, etc., because people in the rest of the United States often don't know what it looks like. But y'all are obviously familiar. I saw papaya plants outside. So when I was told that I was going to be working on papaya, the first thing I did was go Google images papaya plant, right? Because I came from a temperate horticulture background. I knew what the fruit looked like, but I had no idea what the plant looked like. And so the first thing I wanted to know is, what does it look like? Then from there, I went and I started to look at some of the academic literature. It's always nice to start with reviews to kind of give you a broader picture. But the very first thing that I read about papaya was this: papaya has never been found in the wild. Now I thought, what in the world did we do to this species that it had never been found in the wild, but is found in cultivation? But then as I started to get a little bit more into ecological literature, I found all these studies like this, a case study into the effects of domestication. So obviously there was a wild plant involved in that case study, comparative ecophysiology with wild and cultivated plants, and then just pure ecology about habitat and microclimate. So the reason I'll get more into why that matters for horticulture in a little bit, but first I'll go into what the actual ecology of papaya is like. So this is basically a map of the center of origin right here. Papaya is thought to be native to the southern end of the Gulf of Mexico and the Yucatan Peninsula. And the specific microclimate to which it's native is basically this: gaps in neotropical forests, humid or semi-humid forests. This is a wild papaya plant. Obviously that kind of fruit we can't eat, but I just show it to basically show, well, it's growing on the edge of a forest, and that matters in terms of its biology and what kinds of adaptive processes it's gone through as a species. So in terms of production, papaya is the third, or some people say the fourth, most produced global tropical crop. It's an important source of vitamins, particularly carotenoids, in human diets throughout the tropical world. And it's produced in both large plantations, like what we generally purchase here in grocery stores was usually produced in these large monocultures, but also throughout the tropical world is also produced in smallholder plantings. Some of the production challenges that they face in Florida in particular are papaya ring spot virus. So this would be a healthy leaf right here of papaya plant, and this would be a papaya ring spot virus infected plant in my planting. Another important challenge that they face is wind damage. So this picture here is about a 1-year-old papaya planting in Homestead at the research center there. And this was after Hurricane Wilma. So obviously very high winds are a major problem, but also on a regular basis, either mild tropical storms or just the typical windy thunderstorm can cause damage to especially the young plants. So in response to that, we said, well, what can we do to reduce wind damage? One of the answers was, of course, well, you put up a barrier. Some tropical crops are actually, like carambola, are generally grown with netting wind barriers, but given the economics of papaya, that was too expensive, basically, for papaya growers to put in. So we thought, well, maybe we can do some kind of intercrop. And the first one that we decided to test was sun hemp. The species name is Crotalaria juncea. It's a very erect, very vigorous tropical or subtropical legume. So obviously there were a number of potential benefits there. One was it would grow fast enough and dense enough to actually block the wind. Another was, well, you're getting a source of organic matter and nitrogen that you can add to the planting. So in terms of thinking about if you're going to try to intercrop this cover crop, the sun hemp cover crop, you're going to be experiencing a couple of things. And so we looked into the biology of papaya as it applied or pertained to an intercropping system. One important characteristic is that papaya are very sensitive to vapor pressure deficit, possibly a connection also to their sensitivity to even mild, even mild winds in terms of producing a physiological stress. So higher humidity is basically going to lead to higher rates of photosynthesis and growth. So we thought that would be a positive for growing in an intercropping system. They're very sensitive to wind, so we block the wind. So another positive point. In terms of photosynthetic acclimation, they're certainly capable of acclimating to the shade. And this is a shot from a study that was done in 1992, I believe. And this is a full sun leaf, and these are leaves of varying qualities of high shade. And these plants were grown for months under 98% shade and were capable of growing. So we say, well, they're certainly capable of acclimating to high levels of shade. But what happens when you remove that shade, right? So generally our assumptions are that if you have a plant that's growing in the shade, if you have a shade-adapted plant, so a plant that's adapted to acclimation to shade, you have a high level of growth, you remove that shade, and you go to a very low level of growth, and then at some point it's able to reacclimate to a lower level of growth. Something similar in a sun-adapted plant, if it acclimates to the shade, it's not going to grow as well as it does in the sun. You remove that shade, it undergoes a stress, and then it's able to reacclimate. We weren't really sure where papaya would fall in terms of this reacclimation and stress process. But before I go into that, I just want to go into the physiology of high light stress, which this is the physiology of high light stress, basically. Photosynthesis is this amazing process where a huge amount of energy comes in and is absorbed by a leaf. And it reminds me of, if y'all saw the movie, the first Back to the Future movie, where, you know, that scene where Marty's going to drive the car underneath the wires and the lightning's going to strike at exactly 12:04 or whatever. And so, you know, you're watching that scene and you go, okay, well, either something amazing like time travel is going to happen, or, you know, Marty is going to end up looking like Doc. And so that's basically what happens with plants, right? You have a huge amount of energy coming into the leaf. It's going to be absorbed, so either something really cool is going to happen like photosynthesis, or the leaf is going to fry. So basically figuring out how papaya deals with that change in incident energy load was important for for this system. So one of the things that we considered was, well, is there any way that we can alleviate or prepare the plant for the high light stress when we mow down this intercrop and use it as a mulch? And so we turned to this hypothesis of priming. Primed acclimation is a relatively recent application of the concept of priming that's been demonstrated in a couple of agronomic crops, but hasn't been done in fruits crops. And so basically, priming is any treatment that you can give a plant that improves its subsequent or its response to a subsequent stress, right? And that can be biotic or abiotic. But primed acclimation is specifically using regulated deficit irrigation to induce that priming effect. And so far, it's only been used in terms of preparing the plants for subsequent drought stress. However, there are some common mechanisms between water deficit stress and high light stress, and so we thought, well, maybe we can prime these plants using water deficit, which is relatively easy to control, and actually improve their response to high light stress, which we called cross-priming, which we thought was very cool at the time. So this is basically the system that we're looking at. We thought, well, we have a couple of positive things. We'll have a wind stress reduction, reduced risk of wind damage. We'll have this green manure to mulch onto the papaya beds. Papaya is capable of acclimating to shade, so that's a good thing. But we'll have this high light stress, and we may have excessive shading. So the papaya may be able to grow under these conditions, but we may have reduced growth or reduced yield if the shade is too intense. And so we add this concept of cross-priming, trying to find a way of mitigating that high light stress. So overall objectives were to test the use of the sun hemp intercrop basically as a production system in terms of what are its effects on yield and growth, and also to test the use of primed acclimation to mitigate the negative impacts of the system or that we assume will come in the system that we're testing. So we implemented a number of studies to do this. We started with a soil dry down, and part of the challenge in doing water deficit studies is that many times they're very subjective in terms of the levels of the treatments that are imposed on the plants. So what we wanted to do was try to come up with a plant-based approach to developing our priming treatments. So we did a dry down study in order to determine, well, how dry does the soil need to be in order for us to produce a measurable plant response without greatly reducing growth and photosynthesis. And then we went from there to test that in a shade priming study, basically test that priming treatment that we determined based on our dry down study, and test priming as a treatment for reducing high light stress. Then we went on to see how long the priming effect lasts and basically test it against actually a severe water deficit stress as opposed to a high light stress. And then, of course, take that out into the field and test the sun hemp intercrop and mulch system as a production system. So just very briefly, the dry down study, I'm just going to show you this one slide and move on to the other studies. But basically, this is stomatal conductance, so how open or closed the stomata are, which was the most sensitive variable that we measured during the dry down study. And this is that vapor pressure deficit response. So this is high humidity over here above the leaf, and this is low humidity over here, or relatively low humidity over here above the leaf. And of course, you have a strong response of stomatal conductance to that. But then this is the soil section. So this is drier soil to the right, and this is a saturated soil at 0. And so basically, what we were able to do is say, well, at about minus 30 kilopascals in terms of soil water suction, that's a place where we can measure a stomatal response, but photosynthesis was not greatly reduced at that point. So we would expect the plant to have some kind of a priming response if you kept the soil at that soil moisture level. So we took that and we said, well, we're going to test this plant-based approach to priming based on the dry down study, and then also determine whether that priming treatment actually alleviates photooxidation or light stress when we actually expose shade-acclimated plants to full sun. We were also not really sure in terms of the timing of our treatments the priming treatments relative to the stress. So we imposed 2 different timings of drought stress. Oh, it's shaking. So basically our treatments we had no shade. That's the plant that you see out front here. It's just in a pot. Basically these bags were basically put over the plant in order to keep the rain out. And then we had shade structures over half the plants. That's a 50%, 50% shade in an acclimation period of 3 months. And then in terms of priming treatments, we had, of course, a no priming control. Early priming ended a month before we removed the shade, and late priming ended just 2 days before we removed the shade from these plants. And that priming consisted of 3 weeks of watering every day to that target of minus 30 kilopascals. We measured a number of different variables, leaf gas exchange, chlorophyll fluorescence, and I'll explain a little bit about chlorophyll fluorescence. Basically, when light comes onto a leaf and is photosynthetically active light is absorbed by chlorophyll, there are a couple of different ways that energy can travel. It can basically be moved into the electron transport chain, or it can be dissipated as heat, or it can be refluoresced at a lower wavelength. And in many physiology studies, we use that, we manipulate the light environment and use that fluorescence signal in order to measure characteristics of the photochemical side of photosynthesis. Generally speaking, historically, it's been used basically just to say, well, photooxidation, light stress, how functional are our reaction centers. But there's a newer method that we tested here that basically it uses kind of like an echo in order to determine, well, not just how much energy goes through here, but where does it kick back? So where are the bottlenecks in this energy chain here? All the way up to just before photosystem 1, which is basically the beginning of real photosynthesis. So in terms of our results, this is a variable over maximum fluorescence. It's basically a measure of functionality of our photosystems. And if we have a light stress, basically we would expect a level below 0.7. And you can see here, this is our different priming treatments, and full sun and shade are the 2 lines across each one. And that gray bar represents when we remove the shade. And basically what we saw was we didn't provoke a light stress at all. So we acclimated these plants to 50% shade for 3 months, fully acclimated, removed the shade, and we are not measuring any light stress. So obviously we decided we need to rethink that assumption about light stress in our field study. All those things that we thought we knew as physiologists weren't necessarily true in this case. But we saw some very interesting responses to the priming treatment in terms of photosynthesis. So this is leaf level photosynthesis, so this is measured in terms of actual carbon fixation. And it's just set on a relative scale to make it more visually readable. And basically what happened was after we did our early priming period, we were planning on starting measuring out at this point here, but after our early priming period, we went out there and said, shoot, we're seeing greener leaves and this amazing response. We better measure what's going on in terms of photosynthesis. So we went out and started measuring here and found a pretty significant increase of leaf-level photosynthesis in response to the priming after the priming had been relieved. So these plants are now being fully watered. We saw a similar response. So in the late priming treatment, basically halfway through, no reduction in photosynthesis. At the very end, a slight reduction in photosynthesis, but as soon as you restore full irrigation to those plants, they're photosynthesizing at about 30% more than the control. However, over time, it seems like there may be some convergence, like it sort of subsides back to whatever that control level of photosynthesis is. So when we took a look at our chlorophyll fluorescence, our OGIP measurements, basically this is a measure called driving force, and it's based on chemistry, basically, that says this is essentially the ease of reaction, the ease at which the photochemical side of those reactions occur. And that late priming treatment in those days, immediately after that late priming treatment, you have an increase in the photochemical side of photosynthesis, not just in carbon fixation. So he said, okay, well, what's going on with our photochemical apparatus? And this is that same schematic here. So here's where the light's coming in and being absorbed, and then it moves through to the reaction center and then on to a couple of other steps in the electron transport chain. And basically what we see here is that in that primed plant, the primed plant here, dissipation and the amount of energy that actually makes it through to PS1 are about the same. In the not unprimed plant, basically more energy is dissipated than actually makes it all the way through the photochemical side of photosynthesis. And that was all due to changes, sort of downstream changes. So basically opening up those bottlenecks. So if you're going to compare those to actual bottles, basically an unprimed plant is kind of like a Coke bottle, and the primed plant is kind of like a Snapple bottle. So if your goal is to get all that liquid out as fast as you can, you want a Snapple bottle. And that's basically what's happening with these primed plants. So we're thinking, well, there is promise for this hypothesis of cross-priming. There may be an effect on light stress. However, some unique things are going on with papaya that seem to allow it to avoid high light stress, even though it's shade, or it appears to be shade acclimated. One of the things, though, that we weren't sure about is, well, how long does this last? Seems like most of those measurements, the primed plants began to sort of converge with the unprimed plants over time. So that's why we went ahead and did this priming memory study. We said, well, for this study, we'll forget about that cross-priming hypothesis, and we'll look at priming and then response in terms of drought stress in order to not confound our stresses, basically. So we had one priming treatment and extreme deficits imposed at 1, 2, and 3 months after that priming treatment was relieved, basically. So the deficit basically consisted of allowing the plants to dry down to minus 40 kilopascals and then allowing it to go 1 week past that. So there's a little bit of variation between the treatments, between the 3 treatments in terms of exactly how dry they got. But minus 40 was, based on our dry down study, an amount that would induce a significant reduction in photosynthesis. And then the priming treatment, basically we went even lighter, basically minus 20 kilopascals, based on what we had observed in the previous study. And we took a number of measurements. I'll just go through just a few of them. So in terms of photosynthesis, we saw something very similar to what we saw in the previous study. As soon as we're done, this gray bar represents the end of the priming period. As soon as we're done with the priming treatment, we see an increase in leaf level photosynthesis. It's still there at about a month after, but by 2 months after, that's 2 weeks, or 3 weeks, and then 6 weeks. So by 6 weeks after, we don't see any significant differences between primed and unprimed plants throughout the rest of the study. And another thing that we found was that in response to our deficit treatments, there was no effect of priming on in terms of its response of photosynthesis to the deficit treatment. So during each deficit treatment, there was a reduction in photosynthesis, but it was relieved basically as soon as the water deficit was relieved. There wasn't a long-term effect of that extreme deficit, and for primed and unprimed plants, the level of that stress, as far as we could measure, was the same. So one thing that happened when we finished the priming treatment was when we began to dry that deficit 1 plants down, the primed plants began to dry noticeably faster than the unprimed plants. And so what we had to do was we had to go out and start watering every day, water those primed plants up to the moisture level of the unprimed plants in order to keep that dry down parallel between the two deficit treatments. And because we were doing that, weighing those pots every day and watering, well, we got measures of daily plant water loss. And so this is our total water loss over that dry down period. Here's and that's by mean soil water tension, just to compare exactly on average how dry those pots were, to make sure that we didn't impose a different deficit treatment between the primed and the unprimed plants. And so basically what we found was like a 2.5 times increase of water use over that immediate period after the priming in the primed plants. So if you look up here, this is immediately after the priming treatment. Many people would probably look at this picture and say, well, these leaves are lighter, possibly a little bit of chlorosis. That's the stressed plant. This is the unstressed plant. Well, this is the plant that was primed the day that it was rewatered, so not really a lot of time to recover. And this is the plant that was not primed. So definitely differences in leaf level chlorophyll. We have measurements, but I won't show those. But what we did measure is that there was an increased allocation to roots during the priming period. So basically what we hypothesize is occurring here is that you have an increase in allocation to roots even though you don't have a decrease in total growth. And then when you rewater, you have all those roots basically ready to take water up, and the plant is able to sort of maximize photosynthesis, maximize transpiration. But again, one of the things that we observed is, well, most of these effects are temporary, and you have a pretty rapid process of reacclimation. By a month later, you don't see noticeable differences. And the same thing with the deficit treatments. Basically, by a month afterwards, you can't see any differences between the plants that had been exposed to severe deficit and those that hadn't. So we go ahead and take that knowledge out into the field and test this sun hemp intercrop system. Based on our inability to find a high light stress when we remove the shade from those acclimated plants, we decided not to try to do this, the cross-priming thing, so we're just focused on what are the effects of this sun hemp intercrop, and then subsequently using the sun hemp as a mulch on the papaya biology, and then, of course, horticultural variables. So basically what we had in terms of treatments, relatively simple. We have sun hemp planted 4 weeks before transplanting the papaya into the field. This is the day that those papaya plants were transplanted. So you can see sun hemp is a very vigorous crop. It was seeded in 4 weeks prior to this photo. And then in both treatments, the sun hemp was planted on the same day, but we mowed them down on different dates because we weren't sure exactly what the optimum length was. And sorry, those are dates, but it should say basically one treatment was mown 10 weeks after the papaya were transplanted, and one was mown at 15 weeks after the papaya were transplanted. When they were mown, then we subsequently put all that plant material on the beds to use it as a mulch. Of course, measured many similar variables to the other studies: stem diameter and length, and leaf level photosynthesis. We also took some measurements to see exactly what's going on in the microclimate in terms of light, shade, and wind. And again, we're measuring, in order to get at this, still we're wondering, well, is there going to be a light stress when we mow this crop down? And so we use the chlorophyll fluorescence measurements to do that. And then also, we're curious, well, are these plants actually acclimating to shade? If they don't undergo a high light stress, then maybe they're just sort of sun leaves living in the shade. So we took specific leaf weight, which is a proxy for the degree of acclimation to shade. And then, of course, in terms of horticultural variables, we measured sun hemp dry weight and fresh weight, bloom times, and I'll explain the relevance there later. We ended up measuring virus infection, and I'll explain why in a minute, as well as weeding time and yield. Oh, sorry. So, the video's not going. Well, basically, I'll just show you the wind data then. Basically what we see here is this is, in order to take these measurements, because you have wind is very gusty and a point measurement at any given time may not actually be reflective of relative differences between outside your planting and in your treatment plot. Basically the way we measured wind speeds was we had one person stand outside the planting in a clear area where they could measure maximum wind with an anemometer and a walkie-talkie, and somebody else stands in the planting measuring the wind speed just above the papaya canopy, and you wait for a moment of constant wind, and the person outside says, "Now," through the walkie-talkie, and then you write down your wind speeds. It's kind of cumbersome, but it did the trick. And that's why we're presenting this in terms of, well, we have out-of-planting wind speed and then the relative, the wind speed in the given plot. So these circles represent the measurements in the sun hemp, or in between the sun hemp, and the diamonds represent the measurements in the control or the no sun hemp treatments. So basically what we have is we have a significant reduction in wind speeds in the planting relative to the control. Just by virtue of being in the planting, the wind was reduced. However, the treatments that had the sun hemp growing on both sides had a further reduction. And just to point out, there have been some studies in terms of like the papaya physiological response to trade winds, which are constant, relatively low speed winds, that found that wind speeds of 4 kilometers per hour were sufficient to significantly reduce papaya growth and as well as yield. And so if you look at that, well, outside the planting, you need a wind speed of about 7 kilometers per hour to get that in the control treatments. You need a wind speed of almost 20 kilometers per hour to get that stress level of wind speed in between the sun hemp. So we think that's definitely a positive in terms of reduction of risk of damage, as well as just providing a more ideal environment for growth for the papaya. In terms of shade, you probably don't need to look at any measurements in order to say, well, yep, they're shaded. But what you see here is, well, there's a gap there, and so when the sun travels overhead in the middle of the day, there's basically full sun, and that's what these measurements show here. This is photosynthetically active radiation over time of day. Basically, early on in the day, you have this is your in between the sun hemp measurements, and this is in the control. You have basically very intense shade early in the day, late in the day. In the middle of the day, you have almost no shade. And this is just the same data expressed as a percent shade. So you have as much as 75% at the beginning and the end of the day, but in the middle of the day, almost 0%. If you integrate that over the whole day, what you get is about 40% shade. So relatively similar to the treatment that we had imposed in pots. Something that we weren't expecting to be quite so dramatic was we had a very dramatic response to mulching, basically. And you can see that, well, that's a lot of biomass that we put on there. I'll show you measurements in a minute. But as soon as these plants were mulched, a couple days later, you can see them greening up, and then you see greatly increased growth in response to that mulch. So these stem cuttings were taken a month after that mown early treatment was mown and mulched, and this is the this is the control that's growing with no shade, no sun hemp, and this is the mown late treatment that's still in the shade at the time of taking these cuttings. You can see that in the back there with all that intense shade. So basically what you see here is that these stems are almost twice the girth of the controls, and it looks like the mown late may even be less than the control. And that's important because stem girth is the best proxy that we have for total plant growth for papayas. And so this is that same data looking at it over the season. So what you see is, the top two graphs are stem height. And these are the 2 years that we implemented this study. And then the bottom two graphs are stem diameter during those same 2 years. What you see is that early in 2014, basically you had elongation before we even started taking measurements of the plants that were planted between the sun hemp rows. And relative to the control, and they remain taller throughout that growth period. You see early on in terms of diameter, not any noticeable differences. However, once again, this is that line for early mowing treatment. So by a month later, you see a pretty dramatic increase in the relative diameter, and then and you see a decrease of the late mown sun hemp. So that's the sun hemp that's still in the shade, right here, relative to the control. After we mowed down this, the late mown sun hemp, well, it also increased and eventually actually even converged with the early mown sun hemp. And both of them stayed at a greater diameter than the control throughout the rest of the measurement period. In 2015, we didn't see the same elongation there. And we did see, it's hard to see because of the scale that I've tried to keep all the scales even across the graphs, but because of the scale you don't notice it, but yes, you do have significant differences in growth here and here and here in terms of diameter. What we think that this is due to is, if you'll notice, the this is the height of the sun hemp here relative to the height of the papaya. This is the height, these shadows represent the height of the sun hemp in 2015. So you notice that the sun hemp was much taller relative to the papaya in the first year, and much shorter relative to the papaya in the second year, and we think that that's what's causing that difference in elongation. You'll notice that the growth of the sun hemp is kind of shaped a little bit differently, and that's basically, we think, a response to different rain patterns. 2014 was one of the wettest years that anybody who lives in Homestead could remember. 2015, we basically got almost no rain until the end of July. So that's why sun hemp is drought tolerant, but it's not magical, so it still doesn't grow when it doesn't rain. So we have less growth, and we end up with less biomass when we're mowing these down. And so we see what we think that less dramatic response to the mulching is due to smaller amounts of sun hemp that we're actually mulching with. So back to that high light stress hypothesis. This is chlorophyll fluorescence. Once again, down below 0.7 would be what we would expect to be a measurement that we would call light stressed. And this is the period when the sun hemp is growing. We mowed it down at this 0 point here. And so what we see here is that, well, we're not seeing any measurable high light stress. But when we look at the leaves and in terms of how acclimated they are to shade, so specific leaf weight is just the density dry weight density per leaf area. But that's our best proxy for degree of acclimation to shade. So this is a mature leaf. This second graph here is a mature leaf at this initial measurement point here. And you see that even after it's what we call a mature leaf, well, you remove the shade and it even begins to converge with a full sun leaf. However, it is significantly less dense in the shade than in the full sun. This is a leaf that was still in expansion when we took the first measurement, and so we were curious as to how that worked in terms of different leaves as they expand. So basically what we found is that at 5 days after mowing the sun hemp down, those leaves that are expanding are indistinguishable from full sun leaves, so they acclimate very quickly. However, these chlorophyll fluorescence measurements that we took were taken on this mature leaf. So even though the leaf is measurably acclimated to the shade, well, it still doesn't undergo any measurable high light stress when you expose it to full sun. So in terms of horticultural results, one of the things that we observed when the sun hemp was first growing was what you can kind of see here in these two pictures. This is a control bed, just the space between two papaya plants, and this is you can see the shadows of the sun hemp there. And basically, we saw fewer weeds. And not being weed scientists, what we decided to do was go out there and measure how long it took to weed these by hand. I went out there and decided to do that. And took a lot of time, but it gave us a good measurement of just differential weed pressure regardless of the weed ecology, the specific species that were growing there. And so what we see here is that in terms of weeding time, there was a big difference between the 2 years. Remember the differences in rainfall. But in both years, both treatments with sun hemp intercrop and mulching, the amount of time that it took to weed these plots was half of what it took for a full sun bed to be weeded. And then in terms of fresh weight and dry weight, we see really huge amounts of biomass in terms of, when you're talking about horticulture. No grower ever thought about hauling this much biomass in on a truck and applying it. So in that sense, we think it's pretty neat. You have a lot of biomass. It's right there. You don't have to move it. Most of the time, if you talk to a grower about mulch, they say, well, yeah, that's nice, but it costs a lot of money to bring in and spread. Well, if you're growing your mulch right next to your plant, it kind of removes that barrier. Another thing is that, so papaya is unique amongst even subtropical crops in that basically it grows until it's vigorous and large enough to flower, and then it flowers and fruits as long as conditions are right for the rest of its life. So earlier flowering for papaya means more time harvesting. And for growers, that means more money. So we were looking at flowering dates. In the first year, slightly earlier in the mown early in both of the sun hemp treatments, but not significantly different. In the second year, we saw 2 and 3 week hastening of flowering in the 2 sun hemp treatments. In 2015, we ran into a significant problem, which was that early on in the planting cycle, we started getting infections with papaya ring spot virus. We saw some infection late in the 2014 planting cycle, but in 2015 they started very early. And so obviously that's concerning because we weren't trying to kill all our plants. And so what we started doing is we go out there every week or 2 weeks, remove the plants that were showing symptoms, and at the same time, we're of course writing it down, and we noticed some differences between the virus infection rates between the 3 treatments. So these 2 gray bars here represent the point at which those 2 different treatments were mown. So the mown early is right at the beginning of this plot, and the mown late treatment was mown right at the end of this plot. And so we think that there was some kind of an infection period right here, excuse me, because we see increasing rates after that. And remember, this is when we actually see the symptoms expressed, not necessarily the moment of infection. And basically, that mown late treatment stayed lower in terms of virus infection throughout the period that we measured, while the 2 treatments that did not have sun hemp growing on either side continued to increase to really unsustainable levels of virus infection. And this is also the reason why we don't have yield data from this repetition of the experiment, because we didn't have enough plants left in these 2 treatments to harvest. So basically, we consider it to be an important result because, well, we don't have yield data, but in this treatment we had yield, and in these 2 treatments we didn't have yield. So, that matters if you're a grower, right? Basically, the overall conclusions were that the sun hemp intercrop system provided basically a native-like habitat for the papaya to grow in. It's very similar to that gap situation to which the crop is native, and that means partial or temporary shade, wind protection, and that transition from shade to sun actually imitates the natural succession to which papaya are adapted in their wild or native habitat. Excessive shading in the 2014 did appear to reduce growth, so that's something like if a grower is considering implementing this approach, they definitely want to be careful about planting dates and heights of sun hemp relative to the papaya they're planting. So they want to plant the papaya and the sun hemp when they're close to the same height in order to prevent the sun hemp plants from shading their papaya crop too much. The sun hemp mulch, however, improved subsequent growth pretty dramatically and hastened flowering, and the papaya evidently are capable of very rapid acclimation to shifting light environments without any risk of reduced growth due to high light stress. Two important other variables that were not initially going to be tested, but it's that weed pressure and virus infection. So it makes a difference in terms of basically shading the weeds, and then if y'all are interested, I can talk about why it might actually be reducing virus infection. So overall conclusions, the sun hemp intercrop has potential. Papaya acclimates very rapidly, but we don't really know what the mechanisms of that rapid acclimation are, or what is the mechanism that keeps a shade acclimated papaya plant from undergoing a high light stress. And it even occurs to me, like, why would it shade acclimate if it isn't going to undergo a high light stress? But that's sort of anthropomorphizing. But in terms of primed acclimation, priming is possible. However, its effects are relatively transitory, and we didn't see an interaction between priming and water deficit stress, although it seems like there may be some promise in terms of high light stress. So I just wanted to go back to, okay, well, papaya has never been found in the wild. I think that making assumptions like these are dangerous for horticultural researchers. And there's a host of information, especially for these crops that are sort of like recently domesticated or still have significant wild habitat. There's a host of biological knowledge that resides in these ecological studies that horticulturists often ignore. And we just kind of start from the assumption of this crop is native to a full sun monoculture in rows, etc. But what we're showing here is that, well, if you imitate the natural microclimate of papaya, you actually get improved growth and potentially improved yield. And then just going back to our assumptions about what this microclimate was going to do to the plants, well, we said, well, if it's a shade-adapted plant, it's going to do this. Sun-adapted plant, you're going to have the decrease and then acclimate to higher levels of growth. And basically what we saw in papaya was something more like this. It just didn't notice. So in terms of future directions, we actually implemented a shade removal study in which we imposed enough shade to induce a high light stress when it was removed, and tested it with primed acclimation, but we haven't analyzed the data yet, so I can't say much about the details of it. And then in terms of the sun hemp system, basically I think that there may be long-term effects of organic matter in terms of how they impact water use efficiency, especially in the low water holding capacity soils of South Florida. And then, of course, there's still some characteristics of that microclimate that might be relevant to look into in terms of how it would affect papaya biology. So moving on to, what would I do then if I were offered the position here? My personal objectives in terms of research are basically these three things. I want to be involved in production system assessment, crop biology research, and how do those two things combine to affect what in agroecology is called the triple bottom line. So that's society in terms of nutritional and food production needs, society, environment, of course. What are the off-production system effects of the system that we're looking at? As well as economics. How are we affecting growers' livelihoods with different kinds of production systems? In this position, I would like to focus on specifically on crop biology. And I would have a couple of sort of organize my goals in terms of short, medium, and long terms. I think that the cup system is very interesting, especially for the specialty crop kinds of scenarios where you're marketing more high-value crops. And I think that there are some physiological reasons to think that, well, that system is affecting nitrogen and water relations in the plant in a way that would actually reduce the nitrogen and water requirements of the system. So I think that would be worth looking into. More recently, there's some work that just came out that looks into the effects of brassinosteroids on on tolerance to HLB. And of course, brassinosteroids are sort of a common stress signal, plant stress signal, and there's reason to believe that primed acclimation and these kinds of abiotic stress primers also affect that brassinosteroid signal pathway. So I think that that would be worth looking into, that abiotic-biotic stress interaction. In terms of the medium term, I would like to look into some of the similar questions that I looked into with papaya in terms of oh, by the way, sorry, this is from that brassinosteroid study. This is one that had the brassinosteroid applied regularly, and this is one that was just infected with HLB with no brassinosteroids. And this is a picture of the cups system here. But when I was looking for pictures of sun hemp in different production systems, I actually ran across this. This is a different Crotalaria species, not Gensia, but a related species. And this is actually from the research center, or at least according to this description, this is from the Lake Alfred Research Center in 1927. So they're doing cover crop research back then. I'd like to know what the results of those studies were, but I think that it's worth looking into planting diversity as well as alternative and additional crops for citrus growers. Long term, I'd like to look more into rootstock management. So my background as a whole plant physiologist, this relationship between above ground and below ground growth and water relations is especially appealing to me. And I'd like to begin to look more into what are, on a characteristic base, what are the impacts of rooting characteristics of rootstocks on scion growth and development. Just to go a little bit into my background, just to sort of emphasize some of the experience that I have. In Arkansas, I had a lead role in 4 funded proposals, as well as other proposals that I wasn't the lead on, but was collaborating on developing. At UF, haven't been as active in acquiring funding, but have helped some collaborators acquire funding and have experience with applications to a number of different funding sources. In terms of research, I've collaborated very broadly, everyone from entomologists to ag economists. And in terms of methodology, I've tried to be as cutting edge as possible. So this is just a picture of one of the readouts from that OJET method, which is relatively new in field applications. And whenever methodologies didn't exist that did the things that I needed to measure, well, I developed a method to quantify root hairs based on digital images, which are very commonly used in field root studies and mini rhizotron studies. Basically, there wasn't a method to quantify root hairs, even though we know that they're biologically relevant, except in citrus, apparently. So we needed to do that for papaya, because this is actually some of what we were observing in our mini rhizotron images of papaya. So we said, well, we need some way of quantifying that, and so we developed a method to do that, which hopefully will be published very soon. And in terms of publications, have been relatively active and productive. I have a lot of experience in extension. In Arkansas, when I worked as a program technician, that was what I was doing, working primarily with blueberry growers, but with a variety of other crop producers basically started a blog, edited large numbers of fact sheets, and developed a production manual for blackberries and raspberries. If you're interested in the kinds of stuff that I've been doing in extension, and as well as like research presentations, you can go to my personal website. It's called growingvincent at .wordpress.com, and you can get a little more information there. And with that, thank you very much for your time.
Questions for Dr. Vincent? I'm sure there's some. May?
But not part of your presentation, but I was always curious. This function of male and female plants, does it come from seed or cultural practices can manipulate? Because everybody wants more female plants.
Right. So the Maridol, most of the Maridol varieties, so that's the kind that are grown in the Caribbean, including South Florida, are female and hermaphrodites, and the solo types which are grown more in Hawaii, are male and female plants. But that's a genetic difference. So it comes from seed. So there's not a cultural practice that can manipulate that ratio, as far as I know.
So can you identify seed? This is going to be a male. This is going to be a female.
There are some foliar genetic tests, like colorimetric tests, but they're relatively expensive. So most growers, what they actually do in South Florida, and I believe this is close to universal in economic production, is they plant 3, depending on the sex ratio of the variety, they plant 3 plants or 4 plants to a hole, grow them out all the way to about 6 months when they start flowering, sex them and cut down everything but the most vigorous one of the sex that they want. It's not an optimal practice, and I have a lot of observations that would indicate that growing them not 3 to a hole would lead to fruit production much earlier, but it is the standard right now. Yes, sir.
Thanks for the seminar. You alluded to possible reasons for why there was that effect on the virus infection. Elaborate on that.
Yeah, so actually when we were getting ready to do this study, I pushed back against my committee in terms of doing sun hemp because I didn't think that it was optimal. And one of the reasons was a study that had just come out of Hawaii in vegetable systems that said, well, sun hemp is highly attractive to PRSV transmitting aphids. And so I said, well, okay, we're growing in PRSV central here in South Florida, and we're going to attract all the aphids to our planting. That doesn't sound like something I would want to recommend to a grower. But after doing that, or after going ahead with that, because my committee said, well, this is sort of just a proof of concept. We just want to see what it is, what the nitrogen relations are and that kind of thing. More studies came out of that same group, and basically what they found was an increase in aphid populations in the sun hemp, but a decrease in PRSV transmission. And so they went in a little bit deeper into that, being a group of pathologists and entomologists, and found that basically the virus is non-persistent in the aphid, and every subsequent feeding on sun hemp led to a reduction of virus titers in the aphid gut, which then led to reduced transmission to, in their case, they were looking at vegetable systems, but the same virus.
Another attribute of sun hemp is that it suppresses root nematode populations. Are they a problem with papaya? Does that have any bearing on?
No, they're generally not a problem in papaya, but yeah, that's been what, in vegetable systems, that's what's made sun hemp, or one of the things that's made sun hemp a desirable cover crop. In fact, in South Florida, another reason I pushed back against my committee was that seed at the time for sun hemp was $4.50 a pound, and no grower was going to plant an acre, of cover crop at $4.50 a pound. But in the intervening time, they found a new source of seed that dropped the price to about $1.50. So by the time I did my planting, every vegetable grower in the Homestead area was doing summer cover cropping with sun hemp. So that was sort of serendipitous. But no, and it's really mostly a problem in some of the vegetables.
Have you just tried, let's call it intermittent priming or stressing to maintain the photosynthesis rate higher as it goes down after 6 weeks?
It's something that we've considered, but it's not something that we've tested. And I don't know of anyone who's tested something like that. Yes, ma'am.
So what's the lifespan of a papaya leaf?
So depending on conditions, about 100 to 120 days.
So would that affect any of your priming memory results, like you were sticking to the mature leaves? So if a leaf flushed before your priming started, would those factors affect your data?
We looked into it a little bit, but didn't see any differences. So what we think that what's going on is a whole plant kind of thing, not relative to the development of the leaf during that particular period.
How many times did you push back against your committee, and how many of those did you win, and how many did you lose?
I think it was just twice, and I think I had a 100% failure rate.
Fair enough.
Photooxidation, could you discuss that a bit more and the significance and importance of it? Why should we be concerned about photooxidation?
Well, based on what I saw here, I don't think that we really needed to be concerned about photooxidation in this particular context. But it's a natural process; it's occurring all the time, and so any leaf that has incident light is going to have damage to those light-absorbing complexes. And basically, if the damage rate is higher than the rate of replacement, then you're going to have a reduction in photosynthesis. And so that's why it's biologically relevant.
But in your papaya studies, you're showing that it's a fairly temporary response, short term.
Basically, we didn't find any significant measurable photooxidation at all. So it was sort of like, we assumed that there would be photooxidation because of the energy involved in the treatments, right? You reduce, this plant is acclimated to a 50% less energy, and all of a sudden you put it at 100%. We said, well, you're gonna have light stress. We didn't even think to challenge that assumption until we started pulling in data, and we didn't see any photooxidation at all.
What other horticultural crops are so sensitive to wind speed?
That is a good question. I don't know of wind studies in many other crops, but carambola is generally very sensitive to wind speed. So that's the one. That one actually is high enough yields and high enough value that they actually construct these giant mesh structures to block wind.
Any other questions? Well, thanks again to Dr. Vincent.